Mitosis is the process of dividing the cell's nucleus and genetic material. It is also known as a type of cell division in which one cell, the mother, divides to produce two new cells, the daughters, that are genetically identical to itself. So basically, mitosis is used for generating two identical daughter cells, where meiosis is a variation of mitosis in which daughter cells receive half the number of chromosomes in the parent cell, and it generates gametes, a reproductive cell, the egg or sperm cells that combine to form an entirely new individual. We start with chromosomes inside of the nucleus. Chromosomes are a thread-like structure of nucleic acids and proteins found in the nucleus of most living cells, carrying genetic information in the form of genes.
First is interphase. This is a portion of the cell cycle in which the cell grows, which is the G1 phase, replicates its DNA, the S phase, and prepares for mitosis, the G2 phase. What happens in interphase is it consists of three phases: G1 phase, synthesis phase, and G2 phase. During the G1 phase, the new cells grow and carries out its normal metabolic activities. The length of how long it can take depends on the cell type. If a cell is going to divide, it moves into the synthesis phase, the S phase. In this phase, the cell spends a considerable amount of time and energy making copies of all its chromosomes. Each chromosome has one DNA molecule that must be copied completely and accurately through a process called replication. In replication, the two strands of DNA separate and each strand acts as a guide for a new strand. The nucleus contains two identical copies of each DNA molecule. These two DNA molecules, as in each pair, do not drift apart and attach to the centromere instead. During synthesis, the cell also makes a copy of the protein complex called the microtubule organizing center. In an animal cell, these are called centrosomes. Finally, in the G2 phase, the DNA has been replicated, but the cell needs to grow more and produce the machinery it needs to for mitosis.
Next is prophase. Prophase is the first phase of mitosis, in which the replicated DNA condenses into chromosomes and the nuclear membrane begins to disintegrate and the spindle fibers form and attach to each sister chromatid. During prophase, the chromatin, a loose complex of genetic material and related proteins that condenses and forms one or more chromosomes when a cell is dividing, condenses into chromosomes visible under the light microscope. Then, mitosis spindle forms. Next, spindle fibers extend from paired centrioles and the centrioles separate. The nuclear envelope dissolves. Finally, spindle fibers attach to the kinetochores, the protein structure where one or more spindle fibers attach to a sister chromatid during mitosis of each chromosome. A few microtubules do not connect with any chromosomes and continue to grow, extending from pole to pole. This extensive network of microtubules make up the mitotic spindle, the dynamic structure that eventually pulls the chromosomes to opposite ends of a cell that is about to become two daughter cells.
We modeled the mitotic spindles by the strings attached to the chromosomes.
Metaphase is the second phase of mitosis. The spindle fibers form and attach to each sister chromatid, and the chromosomes line up in the center of the cell, called the metaphase plate. This is an imaginary plane midway between the two ends of a cell, on which the chromosomes line up during mitosis and meiosis.
Anaphase is the third phase of mitosis, in which the sister chromatids are pulled to opposite ends of the cell. During anaphase, tension from the spindle fibers, or the strings in this case, causes sister chromatids to separate and the chromosomes move toward the opposite poles of the cell. We modeled this by pulling the spindle fibers or the strings through the centrioles.
Telophase is the fourth phase of mitosis, in which the spindle fibers disappear. A nuclear membrane forms around each cluster of sister chromatids and the chromosomes become less condensed. During telophase, nuclear envelopes form around the separated chromosomes. Then, chromosomes begin to uncoil into masses of chromatin. Cytokinesis is the separation of cellular contents that produce two daughter cells at the end of mitosis or meiosis. During cytokinesis, a belt of proteins pinches the parent cell, causing the cell to split apart. In a plant cell, a cell plate, a structure formed during the division of a plant cell that separates the two daughter cells, plays a role in forming a new cell wall.